Now, Aviwe, Avish, Sotsaka. I hope I've got your name right, hon. I don't know which, both these questions, you've sent 2 questions. Both of them are on the same page. So I'm going to start with the first one, and if we have time, we get to the second one. Okay. It says, in certain marine invertebrates, the color of the shell is under the control of one gene with 3 alleles. So what have we got? We got multiple alleles. In different combinations, the 3 alleles produce 4 phenotypes. They are orange, yellow, what's orange, yellow? But orange, yellow, and black. Do you see where this is similar to blood groups, guys and girls? Come on, please tell me that you do. Okay, blood group A, blood group B, blood group AB, blood group O. They are orange and yellow, and black. What do you know? Immediately, we have something very special going on here. This is the same as blood groups. The only thing is that they've now used color of these shells. The table below shows the results of the offspring produced from crosses involving parents of the different phenotypes. Okay, so here's our table. Here we've got the phenotypes, we've got the parents. Okay, that's 1, that's 2, that's 3, and that's 4. Okay, this is your phenotypes of shells. Okay, phenotypes, what we can see. All right, it's what we can see. Genotype in the genes, phenotype, what we can see. And we're looking at colors here. Okay, this is our cross. And this is cross 1, 2, 3, and 4. Here the parents, and here the offspring. Okay, here are the babas. All right. This one is yellow times yellow. And here our kids are, our offspring, are 27 yellow to 9 black. Interesting, as they say in the classics. Dr. Holmes, you know Dr. Sherlock Holmes? Who was his mate? Watson. Okay, so here we go, kind Watson. We have black times black. And what do all the babas look like? They are all black. That means black is something special. And then we have orange times orange. And, oh wow, we have 30 orange offspring versus 10 black. It gets interesting and interesting. Oh, no, more interesting and more interesting. Let's do our English and spokes it properly. Okay, orange times yellow. Okay, and look at this. All orange and hang on. All are orange, yellow. Remember, we have 3, at least 4, there are 3 alleles, okay, that produce 4 phenotypes. And the phenotypes are orange, I'm just putting this here for you, yellow, orange, yellow, which is clearly a combination of the 2, and black. So, when we look at all of this, what can we see here? First of all, it says, name and describe the type of dominance shown here. So, if we look at the kinds of dominance, let me get a different color here. What dominance can you see here? Here are the parents, here are the offspring. When I have a yellow and a yellow parent, all right, what do? They produce yellow and black. When I have two blacks, they are all black. Immediately, I start to think, black is recessive, just like blood group O. Okay, now if I have yellow and yellow, I have 27 yellow and an odd black. So that proves that this is recessive. If I have an orange and an orange, I end up with 30 oranges, which is high. 30 yellows is high. So this is dominant, this is dominant, and again our little black is recessive, our black color. And then here, orange times yellow, and they are all orange yellow. This is the same as our blood group AB. And this one is either blood, let's say that one's blood group A and this one is blood group B. So it's the same as our blood groups. It's the same process. So our first question, what kind of dominance do we see? It is codominance. Okay, and well, it says describe the codominance shown in dominance shown in 4. It is codominance. Why? Because when we cross an orange and a yellow, the two are crossed, we end up with all offspring being orange yellow. Okay, so that's the same as blood group AB. Then question 2 says, which shell is controlled by the recessive allele? It is the black shell. Why? Because look here. Do you remember that our blood groups, we have A, and A is still A. But when we cross it, we've got this little recessive one here. The only time black comes through is when it's black and black, they are all black. So there are no recessive there other than black. And here we have yellows and we end up with 9 black. And here we have orange and we end up with 10 black, which means when we look at them, they are phenotypically orange and phenotypically yellow and yellow, which tells us that must be recessive because you can't see it when there is yellow and you can't see black when there is orange. Alrighty. Use the information in the table to support your answer to your question. Well, I've just done that. When we cross a yellow and a yellow parent, we end up with 28 yellow offspring and only 9 black offspring. And you can't see the yellow in the phenotype. The black in the phenotype. The only time all the offspring are black is when 2 black parents are crossed. Okay, again, when 2 orange parents are crossed, we have 30 orange offspring and only 10 black offspring, which indicates that the gene was present in the orange parents to produce the black offspring, but you don't see, you only see the orange. And then when we cross orange and yellow, we end up with all offspring being yellow. Does that make sense, guys and girls? I hope so. Alrighty, now, I wonder if we shouldn't just do this on a new sheet. Yeah, I think let's do it on a new sheet. Oh, for heaven's sake, come. We're just going to do this. All right, now, the second question talks about a leopard frog. Okay, and this leopard frog can be spotted, okay, or without spots. And to make it interesting, they actually give you the picture of a frog. Right, so if you are scared of frogs, you have a problem. Okay, now it says spotted frogs were allowed to interbreed and they produced 150 spotted offspring and 50 offspring without spots. Okay, now listen carefully. Look, they had spotted frogs, were allowed to interbreed. Okay, so we had spotted frogs and spotted frogs all interbreeding. And what was the result of that interbreeding? We ended up with 150 spotted and 50 without spots. Okay, so 150, the parents were spotted, they produced 150 spotted and 50 without spots. Immediately tells you, just by reading that this is probably the recessive trait. Okay, why? Because if the parents are spotted and if they were pure, then what would you have? Well, they would only ever have spotted babies. But if they were heterozygous, it would result in 50, which is is it a quarter? It's 1/5 that end up being not spotted. So it says which phenotype is dominant. So which phenotype here would be dominant? It's going to be the spotted phenotype. Okay? It has to be. Why? Because when you breed these babies, you end up with a whole lot that are spotted and only a small portion that are not spotted. Okay? So your answer to question 1 is which phenotype is dominant? It is going to be easy spotted. Okay, so it's the spotted, back. It's the spotted back frog. When it has lots of little leopard spots, yay. Okay, question 2, explain your answer. Well, if we just do look, it doesn't even matter doing a cross. If you have 2 spotted frogs, so 2 spotted frogs breed, and they produce 150 spotted offspring. But approximately 50 offspring have no spots. Okay? And, well, that's a 3-to-1 ratio. And the 3-to-1 ratio is when you have heterozygous times heterozygous. Look, I'll do this, quickly do it here. Here's our little Baba cross. Okay, the spotted parents are, what letter can we use? Okay, if we use S's, it's going to be difficult. Let's use capital F and small f and capital F and small f. Be careful when you do a cross to use a letter where the small letter, the lowercase letter, looks the same as the capital letter. Okay, so S's are always difficult. C's are also always a bit of an issue. Avoid them. Use letters where it's different. So A and A, B and B, D's, nice. So F for frogs. Okay, so here we've got spotted, 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 not spotted. So that is your recessive, and it has to be homozygous recessive for that trait to come through here. And that is 50, 50, and here 50, 50, 50 gives you 150 that are spotted. Of those 150, 50 are going to be pure, so therefore homozygous dominant, and you're going to have 100 that are going to be heterozygous. Okay, so 2 spotted frogs, they produce babies that have got 150 spotted and 50 no spots, means therefore that spotted is dominant. Okay? Okay, then a frog that is heterozygous for spotted back was crossed with a frog without spots. So if the frog has no spots, it must be, so this will be P1. Okay, so we go phenotype. So what do we see? We see a frog that is heterozygous spotted times homozygous not spotted. Okay, or without spots. Our genotype is going to be, it's heterozygous, so we were using, they say use the letters D and D. So D and D, okay, times homozygous will be D and D, because it's not spotted, it is recessive. So we have meiosis and we have gametes. And what are our gametes going to be? D, D times D, D. Okay, zip. And our Punnett square. And gametes. And capital D, small d, and we've done this cross 3 times today, people. I hope you realize this. Fertilization occurs, and capital D, small d, capital D, small d, and capital D. No, Kathy, what are you doing? Doffy, doffy, doffy. Da, 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 da. Okay, sorry. Small d, and small d, small d. So our F1 generation, what are we going to have? The genotype, okay, 2 out of 4 are going to be capital D, small d, and 2 out of 4 are going to be small d, small d. This is heterozygous, so that you remember, and this is homozygous recessive. Okay, now our phenotype, what are we going to see? Okay, we are going to see 2 in 4, or 50%, are going to be spotted, and 2 out of 4, or 50%, will have no spots. And there you go. Voila, as they say in the classics. So remember, P1, phenotype. They're going to tell you what mommy and daddy look like. Genotype, what are mommy and daddy's genes? Meiosis takes place. What do we do with meiosis? We have our little gametes. So that's our half plus the other half. We have this, we have that, we have that, we have that, and we have that. Full marks. There you go. Now, people, next week, I want to do dihybrid crosses. And I want to look at things like cloning and what the cloning process is. So if you've got a really nice question on cloning, send it to us, and I promise you I will use it. So we want cloning, we want dihybrid crosses, because we have now covered all the different types of crosses you can get. Okay, we've also done the sex-linked crosses as well, very, very important, and we've done multiple alleles, like for example with blood. Okay, so even like this one that we did with the shells and the different color shells, when they come with multiple alleles, and you've got here the orange and the yellow gives you orange yellow, well, hello, that's your AB blood group. So if you know the basics, you will get full marks. All right, I want you to do summary sets. I want you to practice these crosses. Practice makes perfect. Have an awesome week, guys and girls, because I know that when we are done with this section, you are going to know exactly what you need to deliver for the end of the year in genetics. And let me tell you, to get full marks in genetics is a tick, a spit, and you got it.